All right, everyone. I know we're a minute out, but I have announcements to make, so we're going to start a wee bit early. So hello, welcome to the final talks of today for the Joint Spinach Tagwig 2024 conference. This is session CTRB, Contributed Oral Presentations in Conservation Taxidermy and IPM. This year's meeting was made very possible in part by the generosity of our sponsors, especially our diamond sponsors. We are grateful to them, and we are especially grateful to OK Congre for helping us host such a successful meeting. If you are attending virtually, please ask questions using the chat feature in Zoom, and we will ask the presenters your question for you. We do have two video sessions today at the end of our talk, and our presenters are online, so we can still ask them questions as well. For those of you here, please try to use the microphone so that your question is captured for all audiences. And after the session, please feel free to move the conversation online to the Oxford Abstracts portal. We all have chat rooms set up that will stay active for several weeks. And finally, as a final reminder, by attending our conference, either in person or virtually, you have agreed to abide by our code of conduct, so do that. Now, I would like to introduce our first speaker, Nicole Feldman, Adapting Conservation, Tales from Natural History Museum's Denmark Specimen Restoration. Nicole?
Hi, thank you for the introduction. I'm Nicole Feldman. I'm one of the conservators at the Natural History Museum Denmark. I've been hired alongside two other conservators under the guidance of Bethany Palumbo, and we are going to be preparing specimens for a new museum. It's part of the Natural History Museum of Denmark, and it'll feature 7 galleries with over 3,000 specimens. In preparing scientific specimens for display in the new facility, conservators face the challenges of tight deadlines, limited budgets, and evolving designs and object lists. This presentation will highlight the innovative conservation techniques on 2 specimens, which required creative problem solving and adaptability to address unexpected issues, ensuring that we're ready for exhibition in the new museum building. The new project hasn't been without its challenges. Like any big endeavor, the conservation team has faced a few bumps along the way, and we've learned to adapt on the fly. When we started in the fall of 2022, the exhibition spaces were still being designed, so the team knew that changes were inevitable. It wasn't always clear how many specimens would fit in each showcase or how they'd be displayed. Plus, the object lists for the 7 galleries kept evolving. Some items were dropped due to space, changing importance, or shifting storylines. All these changes led to constant shifts in our timeline and deadlines. The specimens themselves also pose challenges, coming from all sorts of backgrounds. Some have been in collections for centuries and are historically significant. Others have been heavily used in research, and some have already been displayed before. These differences impacted how each specimen needed to be conserved. In this presentation, I'll highlight 2 case studies, an okapi taxidermy mount and a tiger shark skull, to show how we tackled some of these challenges. So if you went to a talk earlier by my 2 colleagues, you will have noticed this okapi specimen already. And now I'm actually going to talk about the treatment. It was chosen for display in the Biodiversity Gallery, and it had been tucked previously away in a dimly lit corner of a diorama at the now closed Zoological Museum. Unfortunately, the condition of the specimen was unclear when selected. Once it was removed, it was evident that this specimen would need a lot of care. So the original mannequin was crafted from plaster, meticulously replicated on a 1-to-1 scale. However, with the passage of time, coupled with fluctuations in temperature and relative humidity, it had taken its toll. The contrasting expansion and contraction between the skin and plaster have led to the unfortunate splitting and separation of the original seams. The evidence of this can be seen in the conspicuous white line running along the body and neck, inside of the legs, and marking the points where the skin is detached and the stitching has given way. As no case designs had been confirmed at the start of the treatment, it was unclear how it would be displayed, what angles would be most visible to the viewer, and whether certain aspects could be hidden. Therefore, a comprehensive treatment was needed to hide these open seams at all possible angles. Having only a few months to complete the removal of the mount from the diorama, documentation, and treatment, it was quickly realized that acquiring fur that would match in color and/or texture to the copy would require too much time. Therefore, I looked to other methods that have been employed by conservators in the fine arts world. A method used by paper conservators and now implemented here involved the creation of faux fur using Japanese tissue paper. For this method, I utilized an archival heat-activated adhesive commonly employed in paintings restoration for canvas lining, and surprisingly effective on taxidermy mounts. The adhesive was carefully applied into the seam gap and refined using a heat spatula. Following this, strips of long fiber Japanese tissue paper were torn, folded, and adhered in place using the heat spatula. The paper was colored with paints, and then tweezers and bamboo skewers were employed to tease the fibers and blend them harmoniously with the surrounding fur. So displayed here are a series of images capturing the before, during, and after stages of treatment on specific areas of the okapi specimen in 2023. Using Japanese tissue paper faux fur helped to streamline the process and yield a visually cohesive result on the taxidermy specimen. However, I was not quite satisfied with the fill. While the fill blended well from afar, the texture and color did not fully integrate into the mount, and depending on how it would be displayed, could be quite visible. With the tight deadlines, any further treatment on the okapi mount was put aside until the final case designs were given to us. So a year later, in June of 2024, the final display designs were sent to the conservation team. The design layout shows that the okapi is raised on a platform as well as a base inside the case, with full view of the specimen from the front, right, and rear. With this elevated height, the average viewer would look up at the okapi in direct view of the open seam running on the underside, front, and rear of the mount. Because of the information on the new case design, it was important to return to the specimen and modify the treatment. Returning to the okapi a year later, it became evident that the Japanese tissue paper fill had changed in appearance. The long fibers that resembled hair began to stick to the base, diminishing a furry texture, and the color had shifted orange. As such, I needed to come up with another fill option. This fill needed to be, again, quite quick, and something that could be applied over the old fill, as it would take too much time to fully remove. Having just previously worked with commercially dyed felting wool as a replacement fur on a large walrus taxidermy, I wondered if this material would work similarly on the okapi mount. To apply, bits of wool were pulled apart to elongate the fibers and then placed over the area of the fill. Using a punch needle and a heat spatula, the wool could be easily entangled with the original fur and the Japanese tissue paper and locked into place by mechanical means only. Here you can see the application of wool along the bottom as I worked my way up. Luckily, the already dyed wool matched very well with the original fur, and only minor adjustments to the color were made to match the subtle striping along the neck. So this method proved to be quite effective and quick. I was able to apply the wool over the open seams in only a few hours. As the color was already close to the original fur, retouching with pigments was also quite quick and straightforward. And so here you can see the before and after, so the before in the summer of 2023 and the final mount after treatment in 2024. The tiger shark skull was also selected to go on display in the biodiversity gallery. So while the okapi mount was once part of a diorama, the tiger shark skull served as a long-standing research specimen, bringing a distinct set of challenges. Despite the availability of other shark skulls and jaws in the collection, the specimen was chosen specifically because it included both the jaw and the chondrocranium, so it was a unique and valuable piece to go on display. The tiger shark specimen arrived in the lab in quite poor condition. The outer layer, made of a calcified cartilage, was cracking, lifting, and in some areas had completely fallen off. Several large pieces were sitting on the base, and this damage was due to prior research, where 7 drill holes had been made through the jaw for scientific analysis. Further, as it had been sitting in storage, a thick layer of dirt and grime had built up over time. Before any treatment commenced, it was important to let the curator and collections manager know that with the addition of cleaning solvents, adhesives, fill materials, and paints, any treatment that I would do would alter its future use as a research specimen. So given the limited time and the fragility of the calcified cartilage, the treatment for the shark skull prioritized stabilizing the cartilage, reducing the lifting, and filling in losses, rather than achieving a seamless, undamaged appearance. This approach contrasts with the Okapi treatment, where the primary objective was to blend in losses so that they were nearly invisible. The lifting cartilage was first humidified to add some flexibility and play to the material. This was done by using a humidification sandwich seen in the bottom photo. The entire setup was wrapped with PVC plastic film to hold it in place for 15-minute intervals. And then I did frequent checks to make sure the cartilage was still intact because it was quite sensitive to moisture. Once the cartilage had become more pliable, the gaps were filled with a bulked adhesive mixture. The fills were sanded down and covered with thick layers of Japanese tissue paper. Sharp tweezers were used to texture the fill to match the surrounding area. And a final thin layer of Japanese tissue paper was applied, toned with paints, and sealed with a thin layer of paraloid B72 to match the sheen of the original calcified cartilage. So this can be seen in the two images with a close-up of the fill on the bottom. The treatment stabilized the cartilage and reduced lifting, but due to its fragility, the cartilage could not fully be set back in place. Rather, by using fill materials and Japanese tissue paper, I was able to prevent any future loss and stabilize. The specimen now appears more cohesive, but still shows signs of its previous use as a research specimen. So in conclusion, preparing specimens for the new museum building does not come without its challenges. In this presentation, I've highlighted the importance of flexibility and adaptability in order to prepare specimens for display, like the Okapi mount and tiger shark skull. This process shows how essential it is to be responsive and innovative in conservation today. And I would like to thank you all for your attention, as well as everyone at the Natural History Museum of Denmark.
Hello. Two quick questions. Number one, do you know why it turned orange?
I have an idea, yes. Do you want me to tell you that one first?
Yeah.
Okay, here we go. So what I think happened is that the kind of fur began to stick to the base, and I had airbrushed on color, and I built it up over time and let it dry. So I'd done orange layers and then built up the brown layers over it. So when it began to stick to the base, the orange became more prominent. I don't think it was fading of the core paints, which is what I used.
Okay, and then my second question is regarding the fill that you had done at the beginning, not the woven one. Why you had chosen the Biva option as opposed to kind of like making fake hair out of kind of Japanese tissue with like Lasco or something like that?
Sure. So I did try multiple things. One of the issues is that it was sunken in a little bit, so I need to build it out and make it flat. And Biva is really great with a heat spatula, and in times like in terms of speed, it was just easier to build up the fill with the Biva and even it out and then apply on top. And it was actually really, really easy to fold strips and just heat activate it onto it. So for time's sakes, that's why I did it.
You can ask it in under a minute. One more question. So we have
Let's turn it on. We do have one question from the virtual. Tanya asks, I'm curious how you all balance specimen research potential with objects being on display and the impact of conservation methods such as you described on the tiger shark skull.
Sure, I figured that might be a question. So one of the things when it goes on display is that we always make sure we have curators' approval, and we make sure that they're aware that any treatment we do will impact future research, and they have to sign off on that. So they're aware that this will no longer be of use as a research specimen. However, there are other options where you can diagram and document where you've done treatment so that they can avoid those areas and use it as research. In this case, it was so unstable that it was all-encompassing, but that was very much made aware to the curators and collections manager.
All right, next up, we have Alice Gimat, and I do apologize if that's not how you pronounce your last name. I should have asked. Okay. How do fishing lines behave in wet collection jars?
Thank you. So I'm working at the NHMD in Paris, so sometimes we have to suspend specimen in jars. So there are several ways to do that, and we might want to use fishing lines for their transparency. But of course, they weren't designed for that, as many of the materials you might want to use. They were designed to be used in water and, of course, short exposure. So we might wonder if they could be damaged by the exposure to the fluids, and especially solvents-based fluids. So it would be a problem for the specimen preservation. So in this project, which was actually an internship project of one of my students, we tried to compare the resistance of several fishing lines and to expose to different fluids. So first, we had to design the experiment. So we have to choose some fishing lines, the fluids, and a way to measure their resistance. So let's deal first with the fishing lines. So there are basically two different types of fishing lines. The first one are the polyamides. You might know them as nylons. And the second one are based on fluorocarbon, so mainly PVDF. So we try to choose at least one of those two categories. And we decided to choose 3 different lines of the same diameter and of the same brand. And we add the first one, so it's a nylon-based one. I will be calling that N sample. We add the fluorocarbon one, which is FC sample. And in between, there is FCN, which is supposed to be a mixture of nylon and fluorocarbon. So now we have the lines, we needed the fluids. So we decided to go for 4 fluids. So water was the reference of our experiments, and we wanted to study the effects of Cailin modified recipe, so Cailin free with, so basically glycerol and thymol. And we also check what would happen in ethanol at 75%, and also formalin 10%, and buffered at pH 6.8. So first, we designed actually 2 experiments. The first one was without any mechanical stress. We just put the line in the different fluids and looked at their properties before immersion and after exposure after several durations. And we had several tests done to it. So we did look at several things, such as first the aspects of the line and also the absorption of fluids on the line. We wanted to see if there was any chemical change of the line after the exposure, but also if there was any deformation or other mechanical properties evolution. And last but not least, we also studied the creep, but it was designed in another experiments, so I will talk to you, we'll detail this a bit later. So first experiment, so here I report the aspect of the line under a microscope only after ethanol exposure. So with transmitted light, we add a small surface change, but the most important was to see if with reflected light on, for example, a blue background, which might mimic the blue plate that we often use in this type of collection, if they were still transparent. And for the first line, the N line, we added some transparency loss, so which was not what we wanted. Now, if we look at the absorption of the fluid, so basically what we did was to weight the line just after exposure, and we did observe a weight uptake for the polyamide lines and the FCN lines. So that might have an impact also on the line resistance, but only for fluorocarbon, we have almost no absorption. Now, no major chemical change was observed, whatever the lines. So that's a good thing. And now regarding the deformation, so we simply measure the length of the line after immersions, and we did observe also for polyamide baseline a contraction, so reduction in length of almost all the sample, and especially the ones in ethanol. Now, what about the mechanical properties? So to test that, we used this tensile test machine, and basically we put the lines between the two jaws, and we will stretch the lines and measure the force, the tensile strength of it. And we will get this kind of curve, and at some point the line will break, and we will get this kind of curve. With several information we can deduce from that, and we can use both information to compare the lines. So we might be interested at the breaking point, and specifically, the maximum tensile strength, also the maximal elongation of the lines, and also with the slope, initial slope, which represents the stiffness of the sample. So let's first talk about the stiffness of those lines. So without any exposure to the fluid, if we compare the three different lines we have chosen, we can see that fluorocarbon lines is less stiff than the two other ones. And after immersion, a short immersion in the fluid, so only 24 hours, there is a loss of stiffness, except for the fluorocarbon lines. And by letting the sample a longer time, so it was 18 days, we did not see any evolution, so no impact of aging inside the fluid. Now, what about the tensile strength? So you might be interested more about the weight you can have at the bottom of the line. So before immersions, we had this kind of measurements with the average, it's not the average, it's a median, it's a line here. And you can see after immersion, for example, in water and in ethanol, but it was similar for the other fluids, that there is almost no evolution, or at least all the measurements are coherent with each other. So fluids do not impact the maximum load after 18 days at least. So last but not least, we did carry out a second type of experiment designed more to mimic what will happen in real exhibition, for example. So we put the line in a jar with a weight, constant weight, and we just took the measurement of their length at different time. And so far, that's a good thing. After 35 days of aging, whatever the fluid, nothing happened so far. So, of course, the experiment is still in progress, and we will see how it evolves. Now, if I sum up a bit what I have told you. So the main thing that changed for the aspect, it was only in ethanol with a whitening appearing for the polyamide sample. We had a lot of absorption for N and FCN. No chemical major change. We had deformation for both polyamide baseline. For the tensile strength at break, it seems to be level compliant, so that's a good thing. Regarding the elasticity, EFC was not the highest young modulus we had, but it was the most stable. And regarding the creep, no change, so that's a good thing for all the lines. So in the end, we might want to choose the AFC because yes, it's more expensive, but it's clearly the most stable ones. But it might be also an environmental issue, so we have to think about that. Because as you might know, there are restrictions now about fluorocarbons, membrane and everything. So might not be the solution at the longer time scale. So maybe another one might be in the future to use threads of nanocellulose, which are transparent and are also strong. So maybe a new idea for designing a new experiment. I will tell you more in the future, maybe. So I want to thank the intern that worked on the project and my colleagues and you for your attention. If you have any questions or if you want to know more about the aging experiments, don't hesitate. Thank you.
Did you see any stiffening? Because I know it was only 18 days, but because you tested the tensile strength and like, did you see it get stiff?
No, not exactly like this, but usually stiffness is defined by the young modulus, so we have access to it. And we... Yeah, that's not stiffness, but no. No, it's not measurable by hand, so you would not feel it as such. It's too small. So no. And most of it is still in progress, so we have still oversample, waiting for maybe analysis in 1 year or 6 months, I don't know yet when we will stop. But since there was no changes, might be well worth waiting longer than I expected, actually, because we had initially initiated the project due to a fall of a specimen in an exhibition. But no one was able to tell me what line they used. So we had to design the experiment from scratch. So we chose something, and so far, those results with this line and this diameter, it seems to be lasting at least 35 days. So maybe for an exhibition, it's enough. Stay tuned. Thank you.
All right, our next speaker is Catherine Leard, exploring ethics in museum pest management.
Hi, everybody. Thank you for coming here today and joining me for this presentation. It is a huge honor to follow so many brilliant people and to learn so much so far. And I'm really excited to talk about how we can continue to explore ethics in museum pest management and what is already ethical. My current position is as the collections care assistant for the DeYoung Museum and Legion of Honor in San Francisco, California. I do want to share, when I put this photo and this background up, my supervisor said that it looked too tropical for San Francisco, especially the Bay Area, where it's very foggy and often very chilly. But we do have a lot of palm trees, and we're in a park, so it does look tropical, but don't let that fool you. So, history of integrated pest management, just a very quick overview. much shorter than I could possibly go on for hours about what IPM is and what it means for museums. This is a quick quote from museumpest.net, where it basically started. There was a huge movement away from pesticidal use in the agricultural industry because it was impacting the environment as well as humans. And there are still a lot of places in the United States today where pesticidal use is very prevalent and can be very harmful to human beings, especially people that live in that area. Today, IPM, I consider it like a toolkit. You can use so many different things to make targeted and effective treatments without pesticides or chemicals, and that includes low temperature, which we learned a lot about from Tom Strang, who actually has been very informative for my work over the years and his work helped us write our own freezer procedure and protocol documents for the museum, and we use that consistently and all the time. Oxygen reduction using gases such as nitrogen or CO2, or species-specific pheromone lures on sticky monitors. If you've seen them around, they're like little triangles, and they allow you to look at routine inspections and also go after specifically pest species rather than things that might be an incidental visitor in your museum. Monitoring those pest monitors as well as areas, cases, things like that, good housekeeping. And my biggest thing right now has been exclusion. So, ethics in museum IPM. Ethics are already there. I mean, we started this movement because we care about the environment and we care about people and their health. And how can we extend that to the beings that we're also impacting still, which are bugs? And I know that there is sort of this idea of like, who cares? They're bugs. But they are living beings, and anyone who's looked at a bug under a microscope on a pest monitor has seen that they do suffer, and it has been proven that they feel pain. And a lot of scientific studies have sort of posited the idea that you should probably use some kind of anesthetic if you're going to experiment on bugs, especially like cockroaches in labs. And so looking at that, maybe we can try and reduce the amount that we actually get on our pest monitors. And that is really basically the whole idea behind IPM. So I think that they really go hand in hand of just adding that additional step of those considerations in a more holistic approach. I really like this quote because it does talk about the fact that you cannot look at pests on a one-to-one ratio with us human beings. That is just not the reality. But we can and we should have a duty to allow them to thrive in their own biotic community. And that is finding ways where they are so happy to be where they are and not in our spaces. It is true that we have to live with bugs. Bugs live on our own faces. Bugs are everywhere. And so what can we do to allow them to successfully meet their utilitarian needs and for them to complete their own evolutionary necessities in their own environment? So here are my sort of ethical considerations that I think are already really well built into IPM when you're doing them within a museum, especially holistically. And I would really pair these things together: prevention and exclusion, interdepartmental collaboration, education, and something new that I've really been thinking about and want to experiment with at my own institution are natural repellents. And you can see an example here. These are door sweeps. Something very practical and cost-effective is like a sandbag, and well-maintained doors, make sure that all the doors are well put together, they're flush with the floor, can really help keep pests out of collection spaces or areas where you do not want them. Prevention and exclusion. So this is really the idea behind it. Prevention, we think about this as like an umbrella, and I like to think of exclusion as the act. So when we're looking at prevention purposes, here's an example of me vacuuming the top of a tapestry. We do this pretty routinely, vacuuming inside of galleries, making sure that we're not getting dust, and that dust is not building up, because dust contains a lot of proteaceous material. It is yummy for lots of bugs in our museums. They love to eat our skin cells and our hair follicles. So we really want to make sure that there's no dust or minimize dust so that it's not delectable to our pest insects. And you can do that through things such as getting rid of dust, but also what we saw on the previous slide, exclusion with door sweeps. interdepartmental collaboration, and education. So I work a lot with our facilities department. That is a longstanding thing that my position has had, and I love it so much because I get to see so many weird parts of the building that otherwise you wouldn't. We do walkthroughs of our emergency pathways, and we check to see how much dust is in there, whether or not our contract pest control company needs to monitor that area, if the exit door is actually properly sealed, if gasketing needs to be replaced, if it should be vacuumed on the outside in order to minimize how many insects are living on the building. And that can be really helpful. I love doing things like that. I'm sorry that that picture is blocking the script, but it's talking about holistic approaches to prevention and collections care. This is an example of me vacuuming actually what is considered a replica chair. So it is not part of our historic collections per se, but it is still under collections care. And so we do vacuum those things, even if they are not historical. And then that is me checking a pest monitor that we would put behind a tapestry to monitor for bugs. And lastly, I want to talk about natural repellents. I learned about this pretty recently, and so that's why there's not as much information on there, but it is something that I want to continue exploring. It is important for modern conservators to record traditional knowledge, identify plants, and apply traditional practices where possible in their own conservation work. This comes from a huge, wonderful book that you can get as a free PDF online, Our Ancestors Knew Best. And it does a survey of Southeast Asian countries and what they have done to add traditional knowledge to their conservation efforts. And I was able to speak with the first UCLA PhD graduate student who did their PhD on using neem leaves to repel carpet beetles. And from her experience, she had seen that neem leaves are used a lot in India, and they are just like everywhere on the campuses as the trees, and then they're dried and put in sachets, and they have no moth problems. They were very confused about how anyone talked about having moth problems. That was not something they dealt with. So that is a potential thing that maybe we could use as well. So looking back at what did people do before we came up with our modern society to maintain and care for their cultural items and their historical collections. With that, I do always want to give the preface of institutional considerations. You have to look at the economic viability of your actions within museum collection and pest management care. So what are you doing that is a risk versus a benefit? Is it worth the risk to try these new things or to be more ethical and considerate towards these beings? I think that there's a balance to be found, especially within each individual institution and how you want to do that. I think I'm very fortunate that I have a lot of buy-in where all of us together want to minimize the amount of pests, incidental, or any kind of insect in general in the museum, and that allows us to really work together to kind of steward this idea and keep it going forward. So this last quote, we ought to refrain from actions which may be reasonably expected to kill or cause non-trivial pain in insects when avoiding these actions has no or only trivial costs to our own welfare. So there are reasons why we do the things that we do. They're very valid. And with that, not a but, an and. You can think of these things as the way that we also think about a lot of other animals and the world. These are some questions that I have about exploring things further for everyone else, because I'm coming here really to ask about what are your considerations? Have you explored ethics at your own institution for pest management? What are your alternative ways potentially to sticky traps? Is there a way that we can minimize incidental visitors, such as spiders and crickets and things that end up being food for pests, but also are not a danger to our collections, and they're not a danger to us. They're good for the environment. How can we get them to not die on our sticky monitors? And what is your threshold? Are you okay with some spiders in your collection? You know, is it something that we would be kind of okay with at some point in the world, because it's not realistic for everyone to be able to keep all of the world and animals out of their collections, especially in places that are not as microclimate controlled or as high-tech in facilities, especially in North America and Europe. And how would you steward your collections and integrate ethical considerations? So these are just things for all of you to think about, and also for me to ask you if you have answers. This is a works cited of a lot of things that I've read over the years, people who have talked about the philosophy behind integrated pest management and such. There is a PDF of the bibliography with a QR code if you want to read it. These are also the works here. And I want to give a huge thanks to Linda Lynn, who is my colleague. She's an objects conservator at the museum with me. And she was a huge inspiration for me to actually push forward and do this talk. I have my good friend Benjamin Kupka and my friend Anna Davis, who helped me edit, and of course Elizabeth Salmon for introducing, for me, a novel concept of natural repellents, and for stewarding me through this and giving me a lot of advice for this talk. And of course, the Corporations of Fine Arts Museums, where I work, and everyone else who's here and has supported me to get here. Thank you so much.
Hello. I don't know who manages your grounds, but have you worked with them in natural ways to prevent, either by planting native plants or things like that, to really try to shelter your building from certain pests?
I'm so glad you asked that. This is something that we just started talking about, and whether or not it would be possible to introduce like neem trees to our campus. They are grown in Florida, and there's some places in California that do grow them as well. Whether or not the National Park Service would be okay with that, this is the next question, but it is something that our facilities department has been open to, and I'm really excited about the idea of it, of us using nature as a way to allow pests to not necessarily want to come in the building, but to like they can stick around outside, like they're happy there.
Hi, thanks for your talk. You talked about collaboration and getting buy-in from other people in the museums. Do you have sort of tips on that? We have, scientists who will say it's not in the job description to check blunder traps and things like that. So any buy-in is a good thing for IPM.
Yeah, absolutely. I think one thing I will say, I am painfully introverted. This is my interaction for like the year. I'm going to go home and lay down with my dog and not talk to anyone for a very long time. But you putting yourself out there, I send a lot of emails to people in other departments. I also annually do an all staff presentation to introduce myself as like the face of like, hi, give me your bugs. And we have pest log stations, so that is a huge thing to get people excited about looking for bugs, people excited to look at their collections. I also collaborate with our conservation departments where we do like annual gallery maintenance and like vacuuming together. So it's like you can see what you're in charge of on display and also help me do gallery maintenance because I'm one person. So that helps a lot with buy-in. It's also just like enthusiastically talking to people about how much you care about what you're doing. And a lot of times you will get rebuffed, but you kind of have to fight through it and be like, I know you don't care right now, but someone else around you is going to care, and then I'm going to convince you. And that really helps. But I would say finding a way to talk with people on a broad scale with your museum, if you can find the courage to talk to as many people as possible, possibly present to your entire staff, and to create areas where it is fun to talk about bugs, and to allow them to not necessarily be scared of them, but to embrace the fact that they are part of our lives and it is part of caring for our collections. We are all stewards, regardless of whether or not you work in our visitor experience department or curatorial, or if you're one of our security staff. We are all stewards here, and you can use that talking point when you're reaching out to people.
This is from Tanya. Have you heard anything about either cedarwood or bay leaves as insect repellents? I have heard about them in past in the past for pest deterrence, but never in context to museum IPM.
Yes, that great PDF of Our Ancestors Knew Best actually has graphics showing different plant material that can be used for IPM purposes, including cedar and bay leaves. And, you know, everyone talks about having a cedar chest for your wool. Cedar works really well. The one thing about it is that it is also an attractant for beetles. So you don't necessarily want to use cedar in your collections if you can avoid it. That's why things like neem leaves, where it kind of is a repellent for all of them. It doesn't kill the pest, it just makes them not want to actually consume the things that are around them because they find it less appetizing.
Okay, our next presenter is virtual, but she is with us online. I'd like to introduce Sofie Gelardi Munksgaard, who's talking about which bacteria are actively responsible for the maceration of bones.
Hello, my name is Sofie Gelardi Munksgaard, and today I'm going to be talking about unraveling the microbial players in bacterial maceration. This was my bachelor thesis, which I did with the help of my supervisors, Rudi Jespersen and Nicole Feldman. In my thesis, I focused on optimizing DNA extraction for comprehensive analysis, and that's what I would like to be talking a bit about today. So, in this presentation, I would be talking about what maceration is. I'll be talking about this experiment setup, the process we went through, and the methods we used, alongside presenting the results we got from it. And I'll be dabbling a bit into future perspective for this experiment. So, what is bacterial maceration? Bacterial maceration is a time-honored and ancient technique utilized for bone cleaning. It consists of anaerobic bacterial agents in a water-filled container. It offers invaluable insights into decomposition processes and microbial interactions. But despite its historical prominence, these microbes in maceration remain a mystery till this day. Previous studies have largely overlooked the microbial component, requiring a thorough investigation to understand the bacterial communities involved. I think every one of us who've done bacterial maceration have noticed that it's not a standardized process, and the results vary from time to time. Understanding these dynamics holds implications for fields like forensic science, archaeology, and ecological research. Also, by understanding these bacteria, we are also opening the door and the possibilities of optimizing and bettering the maceration process and its outcome, and possibly make a standardized method for this bacterial maceration and get a clean outcome every time. So, the setup of this experiment consisted of a hermetically sealed glass jar which contained water. In this water, we had a rat placed in it. The rat has been skinned, gutted, and the larger muscle group has been removed prior to placing it in the jar. When a decomposition takes place, a lot of gases naturally occur, and if we didn't allow them to escape, the vessel would simply explode at some point. So atop of each glass jar, a gas lock has been placed so that these gases are able to escape, but no new air are able to enter, so that we keep this environment anaerobic. I had 10 glass jars in total, 5 of which were placed at room temperature, circa 20 degrees, and 5 of which were placed in an oven at 39 degrees Celsius. We chose these 2 temperatures because we know that maceration can happen both at room temperature and at higher temperatures, what we will call hot maceration and cold maceration. These glasses were monitored throughout the whole process from start to finish. We made sure to photo document the whole process, and samples were taken alongside the process as well. One of these samples we choose to do what is called bacterial isolation on. It's where you take your sample and you grow it on a plate. In this case, a TSA plate. When you have successfully grown your bacteria, you choose a colony and you go and re-isolate them. You grow it again and again and again on newer plates until you're certain that there is only one type of bacteria growing on your plates. We then took our newly grown and isolated plate and moved that growth to a liquid media. This allowed us for better DNA extraction when that time comes. By doing a bacterial isolation, we increase the amount of bacteria in our samples by a lot, and hereby we also increase the DNA yield we can get from those samples when we choose to proceed with the DNA extraction process. So it should not be a secret that this was a lot harder than we initially imagined when we started the project. We have been searching in East and West for the correct way to do it and the best way to do it. And we have dabbled in fields like commercial kits. We have used protocols. We have tried to mix commercial kits and protocols, and so on and so on. And each method have been tested multiple times until we finally found a method that seems to work the best for us. In this case, it was an isolation before moving on to a commercial kit. In this slide, we see the isolation process starting here from our sample to a growing media plate before moving on to the DNA extraction process. And after that, we moved on, of course, till things as PCR and identification of this bacterial species. And by all our different experiments, we were able to conclude that this process is an anaerobic process. It's an anaerobic environment that these bacteria thrive in. As previously mentioned, we did an isolate, so we were only able to look at 2 bacteria species at a time. But these bacteria species we were able to do a DNA extraction on, and we were able to conclude that the species Clostridium and Bacillus are present in the maceration. So we have now been able to conclude it's an anaerobic process, and the bacteria, 2 of the bacteria present are the species Bacillus and Clostridium. For the future, it will be interesting to look at taking DNA extraction directly from the samples. Since we only have the 2 isolated samples, if we are able to do the DNA extraction from a direct sample, we are possibly also able to uncover all the bacterial agents who are present in this maceration. That meaning that if we uncover all the bacteria present, we are also able to uncover what factors are important to upkeep when you macerate. And if we understand them, we are also able to understand the process a lot better. And by understanding the process a lot better, we're able to bettering the process and optimizing it and get a lot clearer results when we macerate in the future. That was all I had to say. Thank you for listening.
So Sophia is available online for answering your questions. If you have questions, please come to the mic and ask them. We do have plenty of time. To get things started, there is a virtual question, unless you want to go. Okay. So, Sophia, welcome. So Tanya asks, you mentioned that you conducted hot and cold maceration. Do you find one produced better results for specimens than the other?
Yes, we did. For the hot maceration, we had the rats at 39 degrees, while at the cold maceration, we just had them at an average living room temperature, so at around 20 degrees. And with the hot maceration, the first rat were done within, I would say, 9 days, while with the cold maceration, it took approximately, I believe, 22 days before they even started the maceration process. So there were an immense difference between the hot maceration and cold maceration. And specimen-wise, the hot maceration is definitely the way to go if you want great results.
You've had enough. I'm just wondering if there is a benefit to using this type of maceration rather than like domestic maceration.
Well, I think I've gotten a lot of questions about why didn't you use enzymes? Why don't you just use domestic beetles and such? And I can't say whether or not one works better than the other at the moment. My focus with my bachelor thesis were to optimize this process. So whether or not there is truly a better way to do it, I cannot say, but I focused on bettering this maceration process. So which process is the best, I cannot answer, unfortunately.
Hi, this was great. So my question is, maybe it's a question of access, so that's exactly my question. Why did you start with something as big as a rat? Was there anything smaller that would make it just easier for you to be able to maybe shorten the process and make it just faster?
Well, good question. I could have used mice, for example, but I have experimented a lot with rats and maceration in the past, and they seem to be of big enough size so I can follow along the whole process, see when they are truly done and such. And so I had enough fluid to take samples throughout my whole process that were a bit of a worry in the beginning that I will run out of liquid eventually, but the rat were chosen both for observation and for sampling.
It was more a comment for discussion with the domestic beetles. So at our museum right now, we don't have any live colonies, and we usually macerate whales and elephants. We don't have, we wouldn't have the size for stuff like that. So obviously for rats, there's plenty of good options. But it's just the whole holistic approach to maceration.
Also, just more of a quick comment. I just thought this was a great presentation. Thank you so much for putting scientific rigor behind museum practices. We do a lot of things in museum prep, and there isn't the rigor behind it. So this is wonderful to hear. And then my comment on the what's best would be composting, but that's a whole nother talk.
We also have a follow-up question virtually. Tanya says, thinks this is really cool. As a follow-up, what do you mean by done? Bones totally clean? How much bone connectivity with cartilage, etc.?
I consider the rats to be done when we're able to pick up the jar, give a little stir, and I can see it fall apart. When the skin lifted off the bones and there were no connective tissue or cartilage holding the rats together, then I would say it was done.
Thank you, Sophia. And that's it for questions. All right, so we have one last presentation for this session. This presenter is also virtual, and I'd like to introduce Rudi Jespersen, Determining the Age of 4 Unknown taxidermized specimens at the Natural History Museum of Denmark.
Hello, my name is Rudi. I am presenting for you today my bachelor's project determining the age of 4 unknown taxidermized specimens at the Natural History Museum of Denmark. The project, I will first present how I worked with my project, and then I will finish off my presentation with my results and a discussion of those results and what to do next. I started out by identifying problematic specimens. I've worked with 4, which I will present on the next slide. And I have then determined or attempted to determine the elements of specimens that may date them the best. These are my 4 specimens. I've got 4 herpetological specimens. There's a chameleon, and a crocodile, and a frog, and an iguana. The chameleon is from Madagascar. It has a museum number, and it has had a museum number since 1853. The acquisition journal attached to the specimen determined the specimen was already old when it was discovered in the collection randomly. And it is described as old and damaged, and potentially from Museum Vomiani or the Royal Collection, one of the earlier registrations of the collection. The crocodile does not have any data attached to it, and it has one label attached in the 2010s. Which suggests that it may be from the Royal Collection. The frog from Brazil has a bunch of data. We know where it's from, when it's from, and by who it was donated, which is exciting. So hopefully you would be able to use this as reference material to cross-reference with other specimens, but unfortunately that was not the case, as you will find out. The iguana, last but not least, has no data at all. They all have some sort of common thread. There is bad or incomplete data attached with them. The frog, not so much, but they all seem relatively old, around 1800s, perhaps reasonably to guess. The historic context for this, collections throughout the ages. In Europe, collections started out as naturalia collections, also known as cabinets of curiosities. They were an obsession among the rich and were usually private collections. Some were available publicly to the less financially endowed, but primarily they were private collections. In Denmark, Olav Worm and the Royal Collection are probably obviously the king had a huge collection, but Olav Worm himself also, being a physician and having ties to the royal family through his profession, had a relatively substantial collection. Historically, the focus was very, very focused on spectacle and shock and awe. Now museums are more about appreciating nature, biodiversity, and education, which is a lot nicer, I feel. The images on the slide, the topmost is the frontispiece from Museum Vomiani Historia from 1655. The bottom one is Museo Cuspiano from 1677. And Cuspiano was an Italian collector. Same sort of person as Olav Worm was. You can see in these images that there are specimens hanging from the walls and ceilings. Materials-wise, there aren't that many. There isn't that much information about what materials and chemicals were used. Mostly it is chemicals, and they are described as preservative chemicals or preservatives or conservation chemicals, and they are mostly pesticides to keep away pests when there was no climate control of any kind. My process is I was looking through potential archival material. I found some material regarding the chameleon, and I found some regarding the frog, both of which I will bring up later. I have taken X-ray photos of the specimens. I was hoping to use the X-rays as jumping off points for comparing them between each other, or comparing them with existing knowledge of historical taxidermy techniques. Unfortunately, that was not the case. They are all extremely different, and they do not really represent any particular sort of taxidermic style. So they were very, very difficult to use these, but it is interesting to see. And the chameleon, I forgot to mention, the chameleon has a little rope at the back, I believe, which was used to hang, potentially used to hang the specimen from a wall or a ceiling, which you saw in the frontispieces. And then materials-wise, I've looked at the stuffing materials of them all. It all seems to mostly be cotton except for the chameleon, which you can see here. This is kapok, and you can see that it is kapok because it is hollow. It has a gas bubble inside of the fiber. And then I've looked up the pesticides, which I've used SEM-EDX for. The topmost you see to the side is from the crocodile, and it's from the crocodile skin, and you can see I've highlighted the arsenic in on this table. There is arsenic present, which means it is likely from after the 1800s, after arsenic soap was, the recipe for arsenic soap was published. The other two tables are from the iguana and the chameleon. The iguana had iron present in its mounting material, and the chameleon had both iron and chromium present in its skin. Chromium in extremely small doses, but still some iron in about the same or a slightly higher concentration than the iguana. I don't know why that might be. I haven't been able to find anything, but that is definitely a noteworthy, I believe, a noteworthy discovery doing this. There are no sources talking about iron in preservatives. Putting all of this information together, the chameleon, I'm determining it based off of the age in the initial acquisition journal number or journal entry, it being old in 1853, and it having having a rope in the back to possibly hang it. It is earlier than 1800s, but based on the archival material I could find of the inventories of the earlier collections, the Museum Bomianum and the Royal Collection, none of the chameleons in those inventories resemble this one. They're European chameleons, and they are not taxidermized or mounted. They are dried specimens. So it is likely not from those collections, although it might not have been described, of course. The crocodile, sometime after the 1800s, I can't get any closer. Maybe if there were some more information about historic evolution of, or historic development of glass eyes, styles of glass eyes, I would be able to say more. I haven't found any. So I can only say after 1800s on account of there being arsenic present. The data remains unchanged for the frog. I did find about 7 different potential acquisition dates for it. There's plenty journal entries about this frog in particular. However, they all have different dates attached to them, so I don't know when it might have reasonably been described in its current state first. And the iguana, I found nothing, so data remains unchanged. I don't know when it's from. Is it possible to determine the age of these specimens? Sort of. I am positive. Although the results were largely inconclusive, I have discovered new information, both information that is not readily found in literature, such as the presence of iron and chromium, which may just be a freak presence. However, also I have found some new information about the specimens, and I've narrowed down the dates of origins from being potentially from 1650 to being sometime after the 1600s, but most likely 1700s for the chameleon and all of that stuff. For further explorations into this, I would love to have a stronger idea of the historical techniques and materials used. And I would also love to have more comparative analysis between known taxidermy pieces with data, going through and comparing them and describing the techniques and materials used in those periods, so you can have a sort of reference data bank for determining unknowns in the same way. I think that would be a huge help, a wonderful tool to have for collections. Thank you all so much for listening. It has been a delight working on this and also to getting to present.
Our presenter, Rudi, is available virtually, so if you have any questions, please come to the mic. Or a comment, come on up.
Sorry, we were just discussing this over there. The iron we have found commonly used in tanning practices, and the chromium as well. So maybe if the tanning was outsourced, I have it in pieces that were sent to a leather worker, and we have iron in it as well. So the iron and the chromium, I think, are both historic tanning processes. Chromium sulfate. From the mid-19th century.
It is possible. I have looked at tanning processes historically. It doesn't seem, chromium at least was not used before 1850s in tanning. I don't know about iron, but it doesn't seem like the chromium would have occurred through tanning unless it is some sort of process that happened later on. It is a very old specimen. It is a chameleon that had the chromium, and the I found one source saying specifically chromium was not used until 1857, but the note, that's why I mixed up the dates, the actual label for it was written in 1853. So there's not, I wasn't able to really narrow it down to chromium, but I won't knock the possibility that it is some sort of material that has at some point been applied to it to potentially treat or help protect the specimen.
We do have a virtual question from Leonard. Is it possible to use radiocarbon method?
It's a difficult question. As far as I and my counselor have been aware throughout this process, radiocarbon dating is very unstable within a couple hundred years from now and into the past. So it would most likely only be possible to date the specimens exactly when they are very old. So maybe the older specimens that I've worked with would be able to undergo radiocarbon dating. You would be able to get some results, but as far as I'm aware, it isn't usually very viable in taxidermy specimens because they are usually only about 200 years old.
All right, well, thank you all to our speakers and to our attendees. Thank you for being here for this entire session. I noticed several of you hung out the entire time. Okay. Also, I'd like to remind you all that links to the on-demand viewing of the session will be made available to registrants. And those are actually being loaded actively. Some are already available, and more are being added as possible. So thank you again to our sponsors, our diamond sponsors, Oki Kongre, for making this conference possible and for making this session, I think, quite a success. So thank you all. Have a great afternoon.
I